Welcome to the TOEFL Listening section. In this part, you will hear one or more conversations in an academic or campus context, such as a discussion between a student and a professor or between two students. You may take notes while listening. Listen to part of a conversation. You will hear it only once, so listen carefully. After the conversation, you will answer questions about what you heard. Some questions will ask you about main ideas and details. Others will ask about implied meaning, speaker attitude, or how ideas relate to one another. Use both what is stated and what is implied to answer.
Good afternoon, Dr. Morgan. Thanks for meeting me during office hours. I wanted to talk about fungal network communication for my seminar project.
Hello, Alex. That is a rich topic. Tell me what aspect of fungal network communication you are most interested in.
I want to focus on how mycorrhizal connections transfer information between plants. I read that it is more than just nutrients moving. But I am not sure what counts as information.
That is a good starting point. In this field, information can mean chemical signals, electrical impulses, or resource flows that change a plant's physiology. Mycorrhizal networks can move carbon, nitrogen, and defensive compounds. Those transfers can alter gene expression in recipient plants, which is a form of information transfer.
So when a neighboring tree is attacked by insects, it can warn other trees through the fungus?
Experiments show that when an attacked tree increases jasmonate-related compounds, the connected neighbors sometimes upregulate defense genes earlier than disconnected neighbors. The signal can be a cascade of small molecules transported along the hyphae, or an electrical change that triggers cascades in the plant.
You mentioned electrical changes. How do electrical signals move in fungal networks?
Fungal hyphae maintain ion gradients and can generate action potential like waves. Researchers measure changes in membrane potential along hyphae that propagate at rates of a few millimeters to a few centimeters per minute. These electrical events can correlate with downstream chemical signaling in host plants.
Are those electrical signals the main way plants communicate through fungi? Or are chemicals more important?
Both mechanisms are important, and they likely interact. Electrical pulses can trigger vesicle release or ion fluxes in the fungus that change transport of signaling molecules. At the same time, small molecules like peptides and phytohormones physically move through hyphal cytoplasm, or the apoplastic continuum, to reach plant roots.
How do researchers show that the fungus is the pathway rather than soil diffusion or root exudates?
There are several experimental controls. One common method is isotopic labeling. A donor plant is exposed to a labeled carbon isotope, such as carbon-13. Later, the researcher samples connected recipient plants and fungal tissue. If the label appears in the recipient, but not in controls where hyphae are blocked, that suggests transfer via the mycorrhizal network.
So labeling is key. Are there physical barriers used too?
Yes. Researchers use mesh or fine barriers that allow solutes to diffuse but prevent hyphal passage. Comparing treatments with and without hyphal connectivity separates hyphae-mediated transfer from pure diffusion. Molecular methods like sequencing fungal communities help too, because they show which fungal species form the common mycelial network.
You talked earlier about species identity. How much does the fungal species matter for communication?
It matters a great deal. Some ectomycorrhizal fungi form extensive long-lived networks that strongly link trees and facilitate large transfers. Arbuscular mycorrhizal fungi often connect herbaceous plants and grasses. The rate and direction of transfer can depend on the fungal partner, its physiology, and its preference for particular host species.
Is there evidence of preferential transfer, for example, towards seedlings or kin?
Yes, there are documented cases. Seedlings shaded under a mature canopy sometimes receive carbon from the canopy tree through the network. Other studies indicate that plants related to the donor receive more resources than unrelated plants, suggesting kin recognition mediated by the fungus or by chemical cues.
That sounds like cooperation. Could networks also spread pathogens or work against some plants?
Absolutely. Networks are conduits. They can transmit pathogenic fungi or viruses, or move allelopathic compounds that suppress competitors. Some fungal species act as strong sinks and may divert resources from one host to another, in ways that are not beneficial to all plants.
What about the so-called wood wide web phrase? Is that accurate?
The phrase is useful for public communication, but it can be misleading. It implies conscious intent and uniform service. In reality, the network is composed of many fungal individuals and species, with varying interactions. Information and resources move, but the patterns are complex and context-dependent.
For my seminar, I want to include methods and 1 or 2 case studies. Can you suggest concise examples I can present?
Pick an isotopic tracer experiment, showing carbon transfer from an established tree to a seedling, and a defense signaling experiment that measured increased phenolic compounds in connected neighbors after herbivore attack. Explain the controls such as mesh barriers and sequencing to identify the connecting fungi.
Could I also mention measurement techniques for electrical signals?
Yes, include microelectrodes placed along hyphae to record potential changes. Combine that with pharmacological blockers to show causality. That will demonstrate both chemical and electrical facets of fungal network communication.
What are current gaps in knowledge that I could highlight as future research?
Open questions include the specificity of signaling molecules, how networks integrate multiple signals from diverse hosts, and the long-term ecological consequences for community composition. We still need larger-scale manipulations in intact forests to measure net effects on survival and fitness.
I am planning to design a small greenhouse experiment. Any practical advice?
Start with a simple donor-recipient pair and use a labeled carbon tracer. Use mesh controls and sequence the fungal partners to confirm connectivity. Measure recipient growth and defense markers over several weeks. Keep replication high, because variability is common.
That helps a lot. Thank you. One last question. How should I phrase the significance in my conclusion?
Emphasize that fungal network communication alters resource distribution and signaling across plant communities. It has implications for restoration, forestry, and understanding plant ecology, because it influences competition, cooperation, and resilience.
Great. I will draft the seminar and send you an outline. Thanks again for the guidance on fungal network communication.
You are welcome. I look forward to your outline and your experiment design.
Listen again to part of the conversation, then answer the question.
If you label a canopy tree with carbon-13 and later detect carbon-13 in a shaded seedling connected by mycorrhizae, then it suggests carbon transfer via the network.
This concludes the conversation portion. For more TOEFL practice tests and study materials, visit Spinta.org.